Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to my channel, Biology Learning Options. So first of all, if you are new to channel, so please subscribe and press the bell icon button. So today I am here in front of you with a new topic, structure of DNA, and introduction to the function and characteristics of DNA. So if I talk about the lesson outline, so basically it includes discovery of DNA, function of DNA, and the characteristics of DNA molecule. So firstly, I will discuss about it. What is DNA? DNA was first isolated by the Swiss physician Frederick. In 1869, he discovered a microscopic substance in the pus of discarded surgical bandages, as it was resided in the nuclei of the cells, so he called it nucleate. DNA, deoxyribonucleic acid, is a polymer that is composed of two polynucleotide chains that coil around each other to form a double helix. It is a hereditary material in humans and almost all the other organisms. Nearly every cell in a person's body has the same DNA. Most DNA is located in the cell's nucleus, where it is called as the nuclear DNA, but a small amount of DNA can also be found in the mitochondria, where it is called as mitochondrial DNA or mtDNA. The information in DNA is stored as a code made up of four chemical bases. DNA bases pair up with each other to form units that are called as the base pairs. Each base is also attached to a sugar molecule and a phosphate group, and together a base, sugar, and phosphate are called as the nucleotide. Nucleotides are arranged in two long strands that form a spiral called the double helix. The structure of the double helix is somewhat like a ladder, with the base pairing forming the lateral rungs and the sugar and phosphate molecules forming the vertical slide pieces of the ladder. So, if I talk about the DNA functions that include DNA in forensic science, mainly the DNA fingerprinting is an essential tool in forensic science. It does not precisely determine the suspect's identity, but helps narrow it down. DNA fingerprinting is also called as the DNA typing, DNA profiling, genetic fingerprinting, genotyping, or identity testing. This technique was developed in 1984 by a British scientist Alec Jeffreys after he noticed that certain sequences of highly variable DNA, known as mini satellites, which do not contribute to the function of genes, are repeated within genes. It is basically a process of determining an individual's deoxyribonucleic acid characteristics. DNA analysis intended to identify a species rather than an individual is called DNA barcoding. DNA profiling is a forensic technique in criminal investigators comparing criminal suspect profiles to DNA evidence so as to assess the likelihood of the involvement in the crime. It is also used in the paternity testing. So if I talk about its processing, that basically includes creating a DNA fingerprint consists of first obtaining a sample of cells such as skin, hair, or blood cells, which contains DNA. The DNA is extracted from the cells and purified. It includes the RFLP technology, which is basically restriction fragment length polymorphism. The DNA was then cut at specific points along the strand with proteins that are known as the restriction enzymes. The enzymes produce fragments of varying length that were sorted by placing them on a gel and then subjecting the gel to an electric current electrophoresis. The shorter, double-stranded DNA fragments were then subjected to a blotting technique in which they were split into single strands and transferred to a nylon sheet. The fragments underwent autoradiography in which they were exposed to DNA probes, which are the pieces of synthetic DNA that were made radioactive and that bound to the mini satellites. A piece of X-ray film was then exposed to the fragments and a dark mark was produced at any point where a radioactive probe had been attached. The resultant pattern of marks could then be analyzed easily, which suspects DNA closely matches the sample from where the crime scene. After the lecture ends, you will be able to answer the questions that what is the name of the scientist who discovered the DNA structure, and you can cite the characteristics of DNA and you can explain the primary function of DNA molecule easily. Now, if I talk about the history or the discovery, so James Watson and Francis Crick in 1953 concluded that the DNA molecule appears as a three-dimensional double helix. And Franklin and Wilkins used X-ray crystallography to study the DNA structure, which helped Watson and Crick with their discovery a lot. The characteristics of DNA, deoxyribonucleic acid, are that it is made up of nucleotide monomers that are the repeating subunits. DNA double helix structure consists of two nucleotide chains. Each nucleotide consists of a sugar molecule, a phosphate group, and a nitrogen containing base, which are basically attached with each other with the help of the hydrogen bonds. Hydrogen bonds between complementary bases hold DNA's two polynucleotide chains together. The information in DNA is stored as a code made up of four chemical bases adenine, guanine, cytosine, and thymine. Human DNA consists of about 3 million bases, and more than 99% of those bases are same in all people. Only the order or sequence of these bases determines the information available for building and maintaining an organism, similar to the way in which letters of the alphabet appears in a certain order to form words or sentences. DNA bases pair up with each other, adenine with thymine and cytosine with the guanine to form units that are called as the base pairs. In the picture, you can see that how adenine bonds with the thymine and cytosine bonds with the guanine with the help of the hydrogen bonds. So, did you know the sequence or order of the bases in a specific DNA strand is related to the expression of specific traits or qualities? So, in the example, as you can see, the sequence GTC-ATT might be responsible for curly brown hair, while GTC-AAT might be responsible for the straight black hair. Functions of DNA includes mainly it stores genetic information that is necessary for an organism to develop, survive, and reproduce. And if there is any damage to DNA, so it can cause cells to stop dividing, cell death, or the cell can be malignant. Meanwhile, it can create a condition of apoptosis and the cancer. So the general summary includes the discovery the double helical model came from the combined work of Edson, Franklin, and the Wilkins. And the DNA characteristics include it is made up of nucleotide monomers. The four bases are adenosine, thymine, cytosine, and the guanine. And the DNA function includes that DNA stores an organism's genetic information. So here is showing an assignment question, that is how does DNA replication occur? Choose a graphic organizer to show the step-by-step process. If you will do this assignment, so you can better understand that what is the structure of DNA and what are its characteristics and functions in general. There is also a famous quote by Sam Kuhn that the genes are like the story and DNA is the language that the story is written in it, so you can better understand it. Thanks for watching this video. Don't forget to subscribe, like, comment and share this video.